So this information is taken from Thermo Fisher cloning. Traditional cloning relies on recombinant DNA methods that begin with preparing a vector to receive and insert DNA by digesting each with restriction enzymes. The digested fragments are then spliced together with an enzyme called ligase in a process known as ligation to form a new vector capable of expressing a gene of interest. This may be the simplest and oldest technique for traditional cloning and laid the foundation for researchers to develop novel cloning methods such as TA cloning, topo cloning, PCR cloning, ligation, independent cloning, and gene assembly that exploit unique characteristics of other modifying enzymes. So let's look at the workflow for traditional cloning. So here you have the vector preparation. And so the issues that you need to consider for your vector are like what restriction digestion you want to use for your vector. You want to look at whether you need dephosphorylation. Perhaps you may need blunt end creation and then purification. Then when you're looking at the insert preparation, this is where you consider what kind of restriction digestion your insert may require, then whether you need blunt end creation, and again, you would the purification method. Then when we come to ligation, this is where you have to consider the T4 DNA ligase, the PEG, the vector to insert ratio is something you have to consider. You have to look at restriction time and temperature, and then purification and heat inactivation if required. Then we're looking at transformation. So the things here that we have to consider are the transformation efficiencies, competent cells that we want to use, and whether we need chemical or electrocompetent cells. Then finally, we look at colony screening. So you may choose blue-white colonies as a form of screening for the colonies that have your insert, or you may look at positive selection or restriction digestion, what type of restriction digestion you need to analyze your insert in the colonies, colony PCR or Sanger sequencing for your screening methods. So vector preparation. So vectors used in traditional cloning methods are based on plasmids, which are double-stranded circular DNAs that replicate inside bacteria independently of the genomic DNA. So all cloning vectors based on plasmids contain a number of crucial elements, including a bacteria origin of replication. This allows it to efficiently propagate within the bacterial host cell. Then single restriction enzyme site or sites, or more commonly, a multiple cloning site that contains a number of restriction enzyme sites to allow ready addition of an insert of interest. And a marker, it can be antibiotic resistance, to select for bacteria after successful uptake of the vector. In some vectors, the multiple cloning site is located within a gene that serves as a marker and permits screening for clones into which the insert has been spliced successfully. For instance, the PUC18 vector, which is a plasmid UC18 vector, expresses the LAC-Z alpha gene, encoding the alpha peptide of beta galactosidase, which, in combination with XGAL, allows color selection of bacterial colonies formed after cloning. The first step in preparing the vector for traditional cloning is to create an insertion site by restriction digestion. The choice of restriction enzymes depends upon the presence and location of their recognition sequences on the vector in the insert and their compatibility for ligation. Vector restriction sites can be found on the vector map or can be mapped using free online tools such as Restriction Mapper. The multiple cloning sites, if available, is often the first choice for insertion, as the region is specifically designed for cloning. After restriction digestion, dephosphorylation of the vector may be necessary to prevent self-ligation, especially if the resulting ends of the vector digestion are compatible or blunt. During dephosphorylation, the enzyme alkaline phosphatase removes the 5' phosphate groups at the ends. This prevents vector self-ligation because the enzyme ligase requires both a 5' phosphate and a 3' hydroxyl to join the two ends in recirculation of the vector. Dephosphorylation of the vector is important to reduce background and favor insertion of the desired fragment into the vector. Both self-ligated vector molecules and insert-carrying vector molecules can be taken up by the bacteria during transformation and will confer the same antibiotic resistance to those cells. This will create a higher background of undesirable colonies if the vector is not dephosphorylated. Blunting of the vector ends may be required depending upon the restriction enzymes used. So here, purification of the desired fragments is recommended for successful ligation. So let's look at the insert preparation. The source of the insert for cloning may be genomic DNA, a portion of another plasmid, or a linear DNA fragment. Regardless of the type of source DNA, a common first step in preparation of the insert is to perform restriction digestion to generate compatible ends for subsequent splicing into the vector. As with vector preparation, restriction enzymes that are suitable for cloning of the insert into the vector are selected. So one of the most popular strategies is to perform double digests of both the insert and vector for directional cloning. So here's an example from Thermo Fisher. So where you've used two enzymes that generate non-compatible ends. So you've used EcoR1 and KPN1. So since the vector and insert ends can join in only one orientation due to compatibility, so EcoR1 with KPN1, this approach allows the insert to be cloned directionally. When performing double digestion, it is crucial that the reaction buffer and conditions are optimal for both enzymes. Therefore, manufacturers' recommendations for double digest reaction setup should be followed. So there may be instances where suitable restriction enzymes are not available. In these instances, the DNA ends created by the chosen restriction enzymes may be blunted or polished for cloning. So blunting will alter the original sequences around the DNA ends. This could cause a frame shift in the gene translation region or disruption within a gene regulatory region. In addition, ligation of DNA with blunt ends is typically more challenging and less efficient than with cohesive or sticky ends. In some instances, a single restriction enzyme may be chosen that cuts both the insert and vector DNA, generating complementary ends for ligation. This method is commonly used in genomic cloning. In situations where it is not possible to use a single restriction enzyme, a pair of enzymes that have different recognition sequences may generate compatible overhangs that can be used as an alternative. So commonly used enzymes for generating blunt ends are the large or clanal fragment of DNA polymerase 1 and T4 DNA polymerase. The choice of polymerase depends on whether the restriction enzyme generates a 3' or 5' overhang. In the case of 3' overhangs, so those generated by KPN1, T4 DNA polymerase is preferred because it has a stronger 3' to 5' exonuclease activity than does clanal. After restriction digestion of the insert and the vector and subsequent blunting and dephosphorylation if performed, the desired fragments can be purified by running the samples on an agarose gel and excising the fragments of interest. Gel electrophoresis also removes enzymes and salts that were present in the digestion reactions. Gel purification kits are commonly available for efficient workflow and reliable results. The kits are based on procedures that use a chaotropic reagent and heat to liquefy the gel, after which the fragments are purified using silica columns or magnetic beads. Let's look at ligation. So once the fragments of interest are obtained, a ligation reaction can be set up to join the insert and the vector. The most common enzyme used for ligation is T4-DNA ligase, which links DNA ends between the 5' phosphate and 3' hydroxyl groups. The T4-DNA ligase reaction requires ATP, DTT, and magnesium, which are generally supplied in the reaction buffer. To improve the outcome of ligation, a general recommendation is to set up multiple reactions with varying insert to vector molar ratios, typically the range of 1:1 to 5:1. For less efficient ligations, as is typical with DNA fragments with blunt ends, the addition of macromolecules like polyethylene glycol PEG is often recommended to increase the effective concentration of reaction component and thus improve the ligation efficiency. In your ligation reactions, temperatures may range from 14 degrees to 25 degrees, which is like room temperature, and reaction time from 10 minutes to 16 hours or overnight, depending on the type of DNA fragments and desired yields. In general, a higher reaction temperature requires less time but may produce a lower yield. Now on to transformation. So transformation is a naturally occurring process in which bacterial cells take up foreign DNA at a low frequency. In molecular biology applications, this process is enhanced and exploited to propagate plasmids inside bacteria that have been made competent or porous for DNA uptake. Competent cells are commercially available for efficient and reliable transformation. The most common approach to prepare bacteria to be competent for transformation is to treat log-phase bacterial cells with calcium chloride. When the chemically competent cells are mixed with the DNA from the ligation reaction and then heat shocked at 42 degrees Celsius, some of the DNA is absorbed by the bacterial cells where it begins to replicate. Different strains of competent cells are available, and the choice is based on experimental goals and downstream applications. For instance, to perform blue-white screening, a bacterial strain with a LAC-Z mutation must be chosen. If the experiment calls for digestion with methylation-sensitive restricting enzymes, the plasmid should be propagated in a DCM-, DAM- bacterial strain. For protein expression, the strain should accommodate messenger RNA stability and translation, as well as high induction of the recombinant proteins expression. In addition, transformation efficiency of the competent cells is an important consideration. Manufacturers provide the transformation efficiency of competent cells in colony-forming units per microgram of DNA, so CFU per microgram, generally ranging from 1/10⁶ to 1/10⁹ CFU per microgram. In more difficult ligation and cloning strategies, choosing cells with the highest transformation efficiency can greatly improve the likelihood of obtaining the desired clones. Another method to transform bacterial cells is electroporation. In this technique, electrocompetent bacterial cells and ligated plasmids are treated with an electrical current that creates transient pores in the bacterial cell membrane for DNA uptake. Transformed bacteria, after heat shock or electroporation, are then plated on an agar plate with an appropriate antibiotic and screened by blue-white screening or another method for colonies that carry the desired plasmid with insert. Okay, so we've got our colonies growing. Now let's look at how we screen them for those with the desired insert in the right orientation. The transformation reaction contains a mix of cells with no vector. Then you have some cells where the vector with no insert was taking up. And then you have situations where the insert alone was taking up. And the successfully ligated vector and insert will also be in some of the transformed cells. Bacteria without the vector lack the antibiotic resistant gene and will not grow, whereas bacteria transformed with the vector with or without the insert survive due to the expressed antibiotic resistance gene. Thus, the antibiotic resistance genes allow selection for uptake of an intact plasmid. So if you want to identify whether the transformed colonies contain an insert, a number of methods can be employed. The most common is the blue-white screening and positive selection. So in blue-white screening, this one you're relying on relies on transforming a bacterial strain that expresses a mutant LAC-Z gene. So blue-white screening relies on transforming a bacterial strain that expresses a mutant LAC-Z gene, which can be complemented with the alpha peptide of beta-galactosidase encoded on the vector alpha complementation. So this is encoded on the vector and it's referred to as alpha complementation. So transformed cells are plated on a growth medium that includes a transcriptional inducer for LAC-Z expression, IPTG, and a chromogenic substrate of LAC-Z. This chromogenic substrate is XGAL, which is a really long name, so 5-bromo, 4-chloro, 3-indole beta D-galactopyranoside. Long name. In blue-white screening, LAC-Z will hydrolyze the XGAL, producing a blue dye and hence a blue colony. When a DNA insert disrupts the vector encoded LAC-Z alpha gene, no functional LAC-Z is formed and transformed colonies are white. Another popular screening approach is positive screening, or positive selection rather, whereby a gene lethal to the bacterial host is located in the multiple cloning site of the vector. Successful ligation of an insert into the lethal gene in the multiple cloning site prevents its expression, allowing only transformed cells with insert carrying vectors to survive. Clever! To more specifically identify or characterize the insert, transformed colonies must be further analyzed, as the results of blue-white screening and positive selection can only provide information about whether an insert is present or not, but not that you've actually got the sequences that you're looking for. So to more specifically identify or characterize the insert. One basic approach is to perform restriction digestion of the vector extracted from the positive or white colonies and examine the resulting banding patterns from gel electrophoresis. So restriction enzymes must be carefully chosen and can be used to confirm the size and orientation of the insert. The presence of the DNA insert can be determined by a method called colony PCR, in which a small portion of the colonies is analyzed by PCR. This method requires PCR primers that are specific to the insert, to the flanking vector sequences, or both, to detect the insert. To determine the orientation of the insert, a set of primers that can detect the vector and the insert in a single reaction can be designed. So let's continue the talk about cloning and look at cloning applications and methodologies. So cloning methods rely on molecular biological processes that occur in nature. The techniques are continually being refined and simplified. Therefore, many strategies nowadays permit cloning of sequences of interest from their sources more efficiently. These cloning strategies include PCR cloning strategies, Subcloning basics, library construction essentials, shotgun cloning and sequencing method. PCR cloning is a method in which double-stranded DNA fragments amplified by PCR are ligated directly into a vector. PCR cloning offers some advantages over traditional cloning, which relies on digesting double-stranded DNA inserts with restriction enzymes to create compatible ends, then purifying and isolating sufficient amounts and ligating into a similarly treated vector of choice. With PCR amplification, this cloning technique requires much less starting template, which include cDNA, genomic DNA, or another insert carrying plasmid. Furthermore, PCR cloning provides a simpler workflow by circumventing the requirement of suitably located restriction sites and their compatibility between the vector and insert. Nevertheless, there are a number of considerations related to PCR primers and amplification conditions, the cloning method of choice, and the cloning vectors used, and finally, confirmation of successful cloning and transformation. With respect to PCR amplification of a sequence of interest, primers must be designed and PCR conditions, so components and cycling, optimized for efficient and specific amplification of the template. Primer design tools are available to bioinformatically evaluate and select suitable target-specific primer sequences for amplification. Ligation requires that either the insert or vector has 5' phosphorylated termini. Therefore, if the cloning vector lacks 5' phosphorylated ends, 5' phosphate groups must be added to the PCR primers during synthesis or by T4 polynucleotide kinase for successful ligation. For PCR optimization, reaction component concentrations, annealing temperatures, and template amounts are of importance. TA cloning and blunt end cloning represent two of the simplest PCR cloning methods. Their choice depends upon the nature of the vector and the type of PCR enzymes used in the cloning. TA cloning employs a thermostable Taq DNA polymerase capable of amplifying short DNA sequences. This enzyme lacks 3 to 5' proofreading activity and features a terminal transferase activity that adds an extra deoxyadenine at the 3' end of the amplicons. The resulting PCR products with 3' deoxyadenine overhangs are readily cloned into a linearized TA cloning vector containing complementary 3' deoxythymine overhangs. While relatively straightforward, the limitations of this method include the length of insert, so you can do up to 5 kilobases, the inability to clone inserts directionally, and the high error rate associated with Taq DNA polymerase. So let's look at blunt end cloning. This one involves the ligation of an insert into a linearized vector where both DNA fragments lack overhangs. Blunt end inserts can be produced using high fidelity DNA polymerases with 3 to 5' exonuclease or proofreading activity. Their proofreading activity improves the sequence accuracy of the amplified products. However, limitations include lower ligation efficiencies when inserting into blunt end cloning vectors and the inability to clone directionally. Ligation efficiency can be improved by incubating the amplicons with a Taq DNA polymerase and DATP in a procedure called 3' DA tailing. In here, you incubate 20 to 30 minutes at 72 degrees Celsius, then purifying the 3A DA tailed products. To further simplify and streamline the cloning workflow, specialized vectors have been developed to place and insert into vector, for example, without using a ligase. One such class of vectors includes the in vitrogen topo-cloning vectors, which contain covalently linked DNA topoisomerase 1 that functions as both a restriction enzyme and a ligase. Compared to conventional PCR cloning vectors, these vectors result in shorter ligation reaction times, for example, 5 minutes, and greater cloning efficiencies, for example, greater than 95% positive clones. Wow. And with a much simpler protocol. Furthermore, directional cloning of the PCR products can be achieved with a specially designed topo vector using a specific primer design. After ligation and transformation into the appropriate competent cells, the resulting colonies need to be screened carefully for the correct insert, as well as its proper frame and orientation for subsequent studies to analyze the gene fusions and/or protein expression. So now we need to get familiar with the term subcloning. So subcloning refers to the moving of one fragment of a plasmid into another plasmid that can serve as a vector. There are a variety of reasons why it is necessary to transfer the fragment of interest into a different vector backbone. For instance, the new vector may possess a specific marker or antibiotic selection or fluorescent expression. Subcloning may also be performed to move a cloned fragment to an expression vector of a more suitable host for this study. For example, bacteria, mammals, insects, plants, etc. To place the gene of interest under a different expression promoter, for example, a constitutive to inducible promoter, or to tag or fuse the experimental gene with another protein or a marker. Whatever the goal of the experiment may be, the two most common approaches to subcloning rely on restriction digestion and/or PCR cloning. Subcloning by restriction digestion is the more traditional of the two methods. In this workflow, fragments from the vector and the insert are double digested with two restriction enzymes that generate sticky or cohesive ends. Since the vector and the insert can ligate in only one orientation, so it's directional, and the digested ends of the vector are incompatible for self-ligation, this is arguably the preferred and most common method among other possible restriction enzyme options. For subcloning in protein expression or gene regulation studies, the selected restriction enzyme or enzymes should allow in-frame cloning of the fragment of interest with close proximity to the start codon as appropriate. A second popular approach uses PCR to amplify the region of interest from the plasmid. The resulting PCR product is then cloned into the desired vector. TA cloning or blunt end cloning methods can be used as described in the PCR cloning section, but neither approach maintains directionality of the insert. To achieve directional cloning, restriction sites that are present in the destination vector for subcloning can be incorporated into PCR amplicons by using PCR primers designed with the restriction sites in the 5' end of the PCR primers. Following the PCR reaction, PCR products are restriction digested, purified, and subcloned into the restriction sites of the vector. There are a few considerations when designing the PCR primers with restriction enzyme sites. It's imperative that the introduced restriction sites are unique and not present within the sequence of the fragment to be subcloned. The restriction sites should also be carefully designed to allow in-frame expression of the subcloned DNA. Although there is no consensus on the optimal spacer sequence, a general recommendation is to avoid sequences that may result in primer diamonds or secondary structure formation. Be strong.